So here is the talk from the B team. The central objective of MetaHIT is search for gut bacterial genes, species, communities associated to chronic disease. MetaHIT is an FP7 project that gathers 13 institutions, has started in 2008. There are logos which many of you will recognize, but most importantly, it has expertise, very broad transdisciplinary expertise, which we felt was needed in order to progress towards our objective. The approach that MetaHIT has is to construct a reference gene catalog of the gut microbes to reveal the genetic potential and to develop a quantitative metagenomic pipeline for gene profiling, to use this pipeline in order to search associations of microbial genes and chronic disease, focusing on two pathologies, obesity and inflammatory bowel disease. And I'll spend my time basically on obesity, although I will, if I have some time, have to say a few words about IBD. Just to introduce very briefly quantitative metagenomics pipeline, it really starts with a sample. We use stool. In this, total DNA is extracted, and high throughput sequencing is used to generate very many short sequences, 30 million in general, multiplied by 2, and mapping these short sequences on a reference gene catalog, which generates gene abundance profiles. And these profiles can be then used for a number of purposes: metabolism reconstruction, ecosystem reconstruction, capturing of genetic variability. But I will focus here on the statistical analysis and diagnostics. I like to draw a parallel between this quantitative metagenomics pipeline and a powerful microscope, which all of a sudden can reveal what is the content of the microbes of our gut. We have published the reference gene catalog last year, which contains about 3.3 million genes, 150-fold more than our own genome, and which captures 85% of abundant gut genes from a cohort of 124 individuals that we have analyzed. That has been done basically at BGI, who raised to the challenge of generating huge amount of sequence, about 200 human genome equivalent, and assembling it into a high-quality reference gene set. There's some technical data about how that process has been done. Importantly, we found that the human gut microbiome is well represented in the reference gene set by projecting on that set sequences from 3 studies: ours from 124 Europeans, and 2 previous studies of smaller scope carried out in United States and in Japan. In all cases, about 80% of the sequences can be projected on the reference gene set, which leads us to propose that this set represents well the whole human metagenome. But we should keep in mind that, for instance, African populations have not been analyzed yet, and that the set is likely to expand nevertheless. Just to attract your attention, mice microbiota is different. We can project only a very small fraction of sequences, which is kind of a warning when translating results from mouse experiments to man. Mouse is a wonderful model, the best we have, but nevertheless, there is a word of caution here. So one of the messages of this description of genetic potential is that human intestinal genes are largely shared in the cohort, each and every one of us has about 500,000 of these genes out of the catalog of 3.3 million. So, of course, many genes are found over and over in different individuals. About 40% of an individual genes were shared with at least 50% of individuals of the cohort. And that led us to suggest that we are all rather similar. A corollary of that is that if you looked at bacterial species for which we have sequenced genomes, they should be shared. So we used quantitative metagenomic pipeline to measure the abundance of bacterial species and found almost 60 species present in basically all the individuals of the cohort. However, the abundance of species did vary broadly among individuals, which is displayed here for the 57 species listed, between 10 and 1,000-fold to simplify across different individuals. So, of course, if you don't have a depth of analysis enough, then you are going to miss some of the rare species in individuals and have impression that different individuals are very different. No, we are all rather similar, but not identical by no means. And that has been first, that led to the observation that we have also published at the same time, that bacterial species co-vary. So this is kind of representation, a network representation of that observation, where different phyla have been colored in the same way. The circles display species, and the size of the circle is proportional to the overall abundance of a species. The lines connect species which co-vary at a given threshold. So what you can see is that there will be people basically which will have low or high amounts of Bacteroides or of Firmicutes or of Actinomycetes yet again, which led us to state that there are particular constellations of bacterial species in individuals. Now that concept has been pushed much further, elaborated, and really shown in a paper that we have just published, which describes the enterotypes of the human gut microbiome. We have reported that there are 3 enterotypes, and that has been done on 3 different cohorts by 3 different methods of investigation. The first was based on a historical technology, which gives excellent long sequences, easy to interpret, where we have data for Europeans, Americans, Asians. These individuals could be clustered in 3 different groups. We then took that beyond that with 85 Danes analyzed by Illumina technology. Once again, the 3 clusters were found, and finally extended to 154 individuals analyzed by 454 from the United States. Lo and behold, 3 enterotypes again. The main drivers of enterotypes are Bacteroidetes for one, Prevotella for the other, Ruminococcus for the third. But even if you suppress the main drivers, these 2 enterotypes are reconstituted again, which indicates that it's not a single species, it's a consortium of species which drives the enterotypes. Similar clustering is observed if functions are used rather than bacterial species. One can think about enterotypes in terms of, like we think about the blood groups. They go beyond a phenotype such as age, sex, geographical localization, likely diet, or health. But we don't understand at present what is the basis of that enterotype. we do believe that they should allow much better patient stratification and therefore aid to develop personalized medicine and nutrition. Let me turn now to the focus of MetaHIT, obesity, which we study using a Danish study population recruited from a cohort which has been established over 10 years ago, and for which there were phenotypes available during all that time. Unfortunately, no fecal samples were collected at earlier times. We cannot use this cohort for prospective studies, but we can refer retrospectively to this data. I should introduce at that point briefly another project, MicroObese, which is coordinated by Joël Doré from INRA. The project was basically constructed as complementary to MetaHIT, where we have a nutritional intervention in France with overweight and obese individuals that follow calorie-restricted diet. And we use quantitative metagenomics at this time at INRA, not in BGI, using solid sequencing. The clinician in charge of this study is Karine Clément. So, to assess the genes of the obese cohort in MetaHIT, we use 177 Danish individuals, 30 million sequences for each, and we map the reads on the reference catalog. A huge amount of sequence once again. Unexpectedly, we observed a bimodal distribution of individuals in terms of number of genes that they had, and looked immediately into French cohort where we found the same type of distribution. There are low gene count people and high gene count people in both. The difference in gene number is 35 to 40%, which is substantial. We looked at the genes that these two types of individuals had. In both, we find about 2/3 of the genes of the catalog. However, the high count gene people have about 1/3 genes in addition, while very small fraction of the genes are found only in low gene count individuals. It appears that these individuals stratify as well by enterotypes. This is quite preliminary. Low and high gene people seem to belong to different enterotypes. So what? Well, it was of interest to look at the phenotypes of these individuals, and Oluf Pedersen finds, who is a clinician responsible for the obesity study in MetaHIT, that low gene people have higher insulin resistance and increased inflammatory parameters. Karine Clément observed the same thing in French. Interestingly, retrospectively, these people have increased their BMI more over past 10 years than the high gene individuals. These individuals have lost less weight during 6 weeks than high gene individuals. We looked at two different ways. We used different ways to assess the inflammatory status of these individuals. One is to look simply at the white blood cell count by splitting individuals by median and ask, above and below the median, is there a difference in gene number? Yes, very highly significant. High white blood cell counts have fewer intestinal bacterial genes. The other is CRP measures, which is consistent, though we don't quite reach the significance here. it's, I think, a question of the numbers of individuals that we have. That raises a question, is low-grade inflammation correlated with lower bacterial diversity? Can we find bacterial species which are associated to high and low diversity? To answer that, we tried to group genes. We found a way to group genes into metaspecies. We call them metaspecies because we never can demonstrate that they are from the same species, although we can validate that. Hypothesis is that the genes from the same species should have similar abundance in an individual because they're in the same genome. As we know that genes from the same species vary a lot among different individuals, we should have a very strong filter to cluster genes from the same species, so we can take a gene or few genes frequent in low gene people and find co-varying genes across the whole gene catalog. Actually, that works very well. This is illustrated very briefly for one of the species where there is a sequenced reference genome. We use a number of genes which are annotated to belong to that species, retrieve by covariance analysis a number of other genes, and can show by BLAST that they all map very well to the sequence genome. We can then use a fraction of these genes, or all of them, but for simplicity, we use about 50 genes as a marker for metaspecies. What does that information let us see? We call this the MetaHIT barcode because it looks a little bit like a barcode. Each column is an individual. Each line is a gene. Colors denote the abundance. The individuals here are ordered by gene number, and it's quite easy to see that this particular species tend to be present in high gene individuals, and this one in low gene individuals. Well, this happens to be one of the rare methanogens from our intestinal tract, Methanobrevibacter smithii. Well, these guys seem not to produce methane, and knowing that in ruminants about 6% of energy is lost as methane, well, maybe these guys who picked up more weight during past 10 years incorporated that methane in their fat. We have heard about Clostridium Voltai. It's interesting that that's an organism which seems to be present more frequently in low gene individuals. Beyond the two metaspecies that I have just shown you, we identified a number more, which are strongly associated with gene number among these obese individuals. The scale is logarithmic. Differences are between 10 and 100-fold. Some species are present, more abundant in high, some in low gene individuals, and for most, we don't have reference genomes. We cannot really tell what they are. However, they tend to be highly diagnostic for high and low gene individuals. For that, we carried out receiver-operator characteristics among the obese Danes, calculated all the linear models, and found, lo and behold, the models which have extremely high explanatory value. Area under curve tends to be almost 1. So our model almost perfectly orders high and low gene individuals. Is that due to overfit? Not likely. We can calculate models using people that were not used in the initial analysis. This is like discovery cohort and validation cohort, and the model works just as well. These models may have a prognostic value, identifying people at high risk for obesity complications, inflammation, insulin resistance. So that reveals to us massive changes of bacterial diversity in a substantial fraction of cohorts in 2 European countries. Bacterial communities are qualitatively different in 2 populations and raises a number of questions. Is it true elsewhere? What are the reasons? And what are the effects on health? Briefly, we search for species not only associated with difference in gene numbers, but also with BMI, comparing high gene obese and lean individuals, collected metaspecies by covariance, found a number of metaspecies correlated to obesity. Are they diagnostic? Just to remind you, 18 validated obesity risk loci of our own genome in a ROC analysis have area under curve of 0.58, barely better than 0.5, which is what you get randomly. 6 metaspecies have much better predictive value, AUC 0.85. They order quite well lean and obese individuals. That's the distribution across the quantiles of our order. Lean are on one side, obese on the other. Maximum accuracy is about 82%. How about overfit? We can use micro-obese data as a validation test, and the same metaspecies discriminate micro-obese individuals as well as MetaHIT individuals. There we have only obese individuals. So we can just add them to the MetaHIT ones and see that across the quantiles, micro-obese and MetaHIT individuals are ordered in a way which is not significantly different. Micro-obese individuals were not used to construct the 6 metaspecies model, so our model works, in a way, it's validated. In a barcode way, you get individuals ordered by BMI. You can see now that the 3 species that are on the top are more frequent among the lean than among the obese individuals, while the 3 bottom species are more frequent and more abundant among the obese individuals. Similar distribution is seen for the micro-obese cohort. What are the 6 species? For some we don't know. For others we have some idea, but not sufficiently precise yet. So we do have a way to diagnose obese people just by looking at their microbiota and the limited number of species. How about ulcerative colitis? That's a cross-sectional study conducted under responsibility of Francisco Guelmer, where we compare patients in remission with healthy family match controls. Found 12 metaspecies associated with ulcerative colitis, 7 less, 5 more abundant in patients. This is illustration in a barcode way for some of them. These 2 species are more often present in healthy individuals than in patients. The reverse is true for the other 2. Are these metaspecies diagnostic? Well, indeed, they are, but they haven't been validated in a second step yet. So I think that we do have evidence that bacterial species are associated to chronic disease, obesity, and IBD. We do not know are the causal agents, contributors, or consequence, but I would say that they are candidates for etiology that should be investigated, indeed. The take-home message. Our other genome has much more variability than the first one. I think that personalized medicine should target it. I have tried to show you the evidence for that during this presentation, but can't resist to show you the data from a study conducted in China, where one of the meta-species of MetaHIT, which we found associated with obesity, is indeed associated with obesity in Chinese study as well, but it's even more associated with type 2 diabetes. So I think it's very timely to look across number of situations, number of pathologies, number of situations, and find out what is the status of microbiota. Of course, that is the work of the whole consortium. I didn't have really time to go beyond touching the surface about that, but it's a wonderful group of people which I had great pleasure working with. A number of people at INRA contributed to the MetaHIT via metaspecies concept. Sean Kennedy runs MetaCamp platform, and I should thank colleagues from the Microbees study, Karine Clement, Jean-Daniel Zucker, Joël Doré, and you for your attention, and Philippe for inviting me to give this talk. Thank you.